And thanks very much for the invitation for this presentation. If you skip one back, thank you. And je m'excuse parce que je veux parler en anglais, parce que mon français c'est tellement terrible. And so I need to switch to English now. And we heard now, as Elise said already, we heard a lot about the biodiversity, we heard about the raw materials, we heard about extractions, and we mentioned a little bit the active ingredients. And of course, you all know, in order to sell the product in the end, you have to have a claim. And in order to show that the claim is actually real, you need some test systems, and there we come into play. And I represent here the TEDD Center for Cell Biology and Tissue Engineering in Switzerland, and the idea is that I show you parts of what we do about the cells testing in cosmetics. And first of all, the organization, I represent Michael Ragunat, who is the head of the TEDD, and we have some collaborations with different kinds of industries and universities, from the ETH in Zurich, from the TU in Berlin, and we have different panels of steering committee and advisory boards to keep up with the latest knowledge in science and to translate this into the testing of drugs and cosmetics. So as you can see, we have a large array of members, and they are from big pharmaceutical companies like Roche and Novartis, up to all different kinds of technical companies like Tecan, and even, of course, then going into the cosmetics with Givaudan, and even small, very small, tiny companies in Switzerland with Xenometrics, where we have specific cell-based assays that I will go into later in the talk. Now I want to show you a little bit the expertise that we have local in Zurich and what we can do in terms of testing. We all had the past decades of testing that was done usually with animal cells and with animals in total. Now animals are banned, testing with animals, but we need to have for the claim some real physiological evidence that it is in most likely human physiologically relevant. And so there are different kinds of activities that we have in terms of bioprinting. And we have a high-end industry-compatible bioprinter to print actually tissue and to try to mimic the human physiology, ready for testing, ready to make the claim of these active ingredients. And some of those bioprinting activities include to print liver for toxicity, not so much interesting now, but also kidney and 3D tubules in the kidney we were able to print with cells. We were able to print muscle cells, and not a very nice picture of skin, but also we try to print skin. Next. And Michael and his group did some very specific tests on 2D skin cell tests. And the goal was to test the compounds and find out to what extent they can suppress lipid peroxidation as one of the problems that the cells are facing. And he made a really nice assay, and you can see here the basic results. So untreated cells and not stressed, they behave and they show this red color. And as soon as they are stressed, the lipid peroxidation, we have made an assay that it is easily visible and quantifiable by showing this green color. And if the cells are treated with a compound that prevents this lipid peroxidation, even though we put the stress on the cells are protected. And this is the kind of assay and the kind of claim that the cosmetics industry requires to show actually the proof, the scientific proof that the compounds, the active ingredients are actually working. And then there is also a skin model, a 3D skin model for corrosive. Of course, it's just a stratum and the fibroblasts are missing, and we are trying to build up. So those are just the keratinocytes, and these are now primary keratinocytes, primary human keratinocytes, to get closer to the human physiology as possible. And then when we add some stress to those cells and some corrosive agents, you can see that it completely disintegrates. Then this is a test I did in my group. So what we try to do is advanced cell-based assays and to have everything based on human, most likely primary cells or stem cells. As this is now quite a fancy topic, especially in cosmetic industry, can we show with the active ingredients a benefit for the skin stem cells or for the hair stem cells? And we are developing assays to show actually this. So this we did for Mibel, this assay, to show testing environmental stress. And the environmental stress there was diesel particulate matter. And it's amazing. I didn't realize before we did this how toxic diesel particulate matter is. Actually, this diesel dust that is in everybody, well, at least in Europe, everybody talks about this now. So when we add a little bit of this standardized diesel, you can buy standardized diesel dust, 200 milligrams, $1,500. I mean, I drive with my car for 1 kilometer and I have grams of it, but it's not standardized. Okay, so we have this standardized diesel dust, and when we add a little bit of this standardized diesel dust to the cells, it's German. It's complete massacre of the cells. They're completely gone. So what we had to do is we had to do a kind of in-between thing, and what we did, we added a layer of collagen to simulate a little bit the kind of a skin barrier. And we added the collagen, and then the toxic compounds could diffuse through the collagen layer, and then we could finally make a nice test. And we did this on induced pluripotent stem cells that we developed, so human cells that we tested, human stem cells with this test. And of course then the idea would be the active ingredient prevents the damage of this toxic diesel dust. Another one, one back, sorry. And another one we have is we do also using report assistance. So we take the human primary tissue and we genetically modify them, that we have a quantitative readout system that we can do and test then the active ingredients. And what you see here is the activity, and this is coming actually from the idea with the assay that Shivador made and put it into the OECD guidelines for oxidative stress. And what we now developed here is a better assay, and to test for the skin sensitizers. The idea is that one can add oxidative stress with the ethylene glycol dimethylglutamate and then see how those cells react on the oxidative stress. And we developed this with primary tissue cells, with human keratinocytes, primary keratinocytes, and we genetically modify them so that they produce an enzyme if they are oxidative stressed. And we had now, as you can see, up a 700-fold upregulation. We can test quantitatively, and as you can see with the arrow bars, very sensitively and very precise. With this, we can now test the active ingredients that prevent the oxidative stress to the skin cells, to the keratinocytes. So the idea is that TEDD is, on one hand, a platform to get connected, and that's why I'm here. And of course, also, we have a physical center of competence at our university, and the idea would be to work with us to analyze your actives. Thanks very much.